Hello, everyone, and welcome to today's live broadcast, Proteome-centric Precision Medicine, Embracing Pathological Diversity.
Today's live broadcast.
I am Brenda Kelly Kim, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences is dedicated to improving the health of people around the world. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life science researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. For more information, please visit www.beckmancoulter.com. We have a few announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker. Dr. Jennifer Van Eyck, PhD, is a professor of medicine at Cedars-Sinai Medical Center, director of the Basic Science Research in the Barbara Streisand Women's Health Center, and director of the new Advanced Clinical Biosystems Institute, where she recently moved from Johns Hopkins University. The Institute mandate is to facilitate application proteomics through medicine and enhance translation of new therapies and biomarkers to clinical use through the application of innovative technologies. Her research laboratory studies the underlying molecular mechanism of heart and vascular disease using a large number of proteomic methodologies, focusing on the site-specific identification and quantification of disease-induced post-translational modification. The underlying concept of competitive post-translational modifications that act as molecular switches that sense and respond to dynamic changes within the cell. Widely regarded as an international leader in the field of proteomics, Van Eyck is best known for developing a number of lab tests to determine the presence of certain proteins or amino acids in patients' blood, which could indicate whether or not they have experienced a heart attack or have heart disease. She earned her Bachelor of Science degree in Biology and Chemistry at the University of Waterloo, Canada, and her doctorate in Biochemistry at the University of Alberta, Edmonton. During her directorship of the Johns Hopkins Heart, Lung, and Blood Institute Proteomics Center, a national heart, lung, and blood institute located at the Johns Hopkins University School of Medicine, Van Eyck also served as director of Bayview Proteomics Group and as professor of medicine in the Division of Cardiology. She has over 210 peer-reviewed manuscripts, reviews, book chapters, and has co-edited 2 books. I will now turn it over to Jennifer for her presentation.
Thank you very much, and welcome everyone. I'm delighted that you've all joined today. So over the next 45 minutes, we're going to discuss, hopefully with questions coming from you, really how we can use proteomics to address this emerging field of precision medicine. It actually isn't an emerging field, but rather precision medicine has been the focus of many research laboratories and many clinical laboratories for a long time, trying to be able to get to really this concept of being able to get the medicine directly to the right disease. So that's precision therapy, when we're trying to understand the pathways of disease and being able to develop drugs that will target specific parts of that pathway, so precision therapy. The other side, of course, of personalized medicine is really the ability to diagnose or prognose an individual's disease based on, in our lab, the proteome. So to do this, the concepts behind that is that we want to be able to quantify an individual's proteome, and that comes from the premise that the proteome that we would be measuring at this moment reflects their past, meaning whether or not the epigenetics, whether or not they have been under more redox conditions, will be reflected in the proteome. So that's the present. And how the present proteome is will, of course, dictate how you will respond in the future, so how your disease will progress, and also, of course, how you would react to a certain therapy. So by being able to quantify explicitly the proteome, we should be able to understand their past and the impact of the past on how they will respond to their drug therapy, and being able to produce both the drugs that will be right on target for those particular nuances, as well as being able to diagnose where an individual is in their disease progression becomes really the centerfold of personalized medicine. So to do this, we believe that we have to understand how the different omics interact, and that's just not metabolomics and genomics and proteomics, but all the perturbations within proteomics itself, so the different post-translation modifications that can influence how different proteins interact with each other. So when you think this way, one of the underlying principles that you have to be able to do is to produce a technical pipeline that allows you to be accurate in your measurement across all the different post-translation modifications, or at least a subset, and have an ability to have it scalable. So being able to do 10 samples to being able to do thousands of samples in the same accuracy of the quantification. So when we think again about precision medicine and having an ability to precisely measure these proteins, the idea behind it is to be able to both intervene to obtain better clinical decisions for individual patients. So, of course, that gets down to being able to get the right disease mechanism, getting the right drugs, and being able to select the patient based on biomarkers. But there's another side that's often not discussed, certainly at the proteomics level, and that's the ability to improve public health. And to do that, we have to start to decide and talk about things like point-of-care devices and screening programs and policies that will allow us to reach out into the unmet populations that require better health care that are currently not being addressed. And so today we'll be talking a lot about being able to find mechanisms to have better clinical decisions in the hospital setting. But also at the very end, I'm going to talk about how there's new technology being invented and being applied that allows us to reach out into the population. So this is an iterative process as we start to move both biomarkers and understanding of mechanisms to technologies that allow us to increase our throughput. And always the quality of the data matters, and having precise measurements of data is really what we're about in our research lab. So you might ask, well, why proteins? And certainly proteins are not the only place to get good biomarkers, and it's certainly very reasonable to mix quantitation around proteins with genomics and metabolomics and other omic fields. But one of the reasons that we particularly like proteins is that you're able to really monitor, as I mentioned earlier, the past of a person, their immediate proteome, as well as what we can predict to be their future response. And that's because proteins have this incredible diversity based on both their isoforms or splice variants, as well as post-translation modifications, which exponentially increase the biological diversity of what one protein can do. So measuring the protein itself is important, but being able to quantify whether or not you have a specific splice variant or whether or not you have multiple post-translation modification really is where we need to be in our field. And protein and post-translation modifications are remarkable because a single protein can have one amino acid residue that's modified by more than one post-translation modification. So lysine is a great example of that, as is cysteine. You can have one amino acid that has competitive post-translation modifications competing for it. There are hundreds of post-translation modifications, many of them which we do not have good tools to be able to quantify, let alone identify. And what's really remarkable is that post-translation modifications can be dynamic. They can be reversible and also irreversible, and the one I'll be focusing on today is an irreversible post-translation modification. It's called citrullination. It's a deamidation of arginine when it's part of a protein. And the deamidation of arginine is irreversible, and it is only through the turnover of a protein that you're able to regenerate the unmodified form. At least that's what we believe today. So we propose that if we're able to quantify proteins and these disease-induced modifications and the induction of new isoforms, perhaps we'll be able to be more specific and be able to get this refinement that is required to be able to get our drugs on target and to monitor what's happening in the cell in response to a drug therapy, as well as be able to differentiate individuals and how they'll respond to certain therapies and how they'll progress in the future. So again, the reason to be doing proteins is that they are really central to the whole transcription, translation, and downstream metabolomics and lipidomics. And due to the modifications, the subtleties of what's happening in the cell should be amplified. So today I'm going to talk about really three things. Why we think the individual's proteotype, the proteins that dictate the phenotype, is going to be important. And we're going to focus in on RA, rheumatoid arthritis, and heart disease, and some potential ways that we're thinking about new therapies and new diagnostics to monitor this. How we can do scalable workflows, and a lot will be concentrated on this, really about automation of sample preparation and assay development. And finally, what is coming down next, and being able to really capture the proteotyping in a continuous patient-centric healthcare screening, trying again to move this out into the larger population to reduce healthcare barriers. So let's start with why. So as I said, the proteotype is a word that's supposed to bring to mind both the concept of the proteome and its impact really on the phenotype. So again, starting with heart disease, it is the #1 killer in the world right now. There is every 1.14 minutes someone is diagnosed with heart failure in the US or in Europe, and there is about a 40% survival rate after 1 year of diagnosis. So we do not really have early diagnostic markers or indicators for heart failure. Women, it is the #1 cause of death in women, and more women will die of heart disease than males. And interesting, with autoimmunity, which is of course enriched in women, things like RA and lupus, you have an increased odd ratio of getting heart failure. So there's something about autoimmunity that is driving an increased risk of having heart failure, and of course that is in a female enriched population because autoimmune diseases such as RA, as I mentioned, happens more in women. So RA, if you have RA, you are at more risk of developing heart failure, and also with stroke, there's a higher prevalence of atherosclerosis and higher mortality rates. One of the interesting aspects of RA is that they produce autoantibodies, and autoantibodies can be induced by protein modifications. So being able to, your body to recognize self, so recognize a protein that should be there, will not stimulate the autoimmune response. However, if you have a non-self-recognized protein, so a protein that's been modified, and citrullination, this post-translation modification I mentioned earlier, is a modification that can induce or has a higher propensity of inducing an autoantibody response. And certainly citrullination of proteins was well recognized and has been recognized in the field of RA at heightening the autoimmune response and being at least one of the modifications that can drive autoantibody response. Citrullination is induced by an enzyme family called PAD, peptidarginine deamidases, and what it does essentially is remove the nitrogen group from the side chain of arginine and essentially makes it go from a positive charge to neutral. Excuse me. On the left-hand side, what you'll see is a Rockman-Meyer curve, where you can see the incidence and survival probability of women and men who have RA, of course, is increased, so you're more likely to not survive if you have RA. So we're going to look at citrullination, the post-translation modification that is actually quite challenging in proteomics to assess and quantify. And we have, when we're talking about proteomics, a series of different methodologies one can use for protein quantification, and I've listed a few here. So in the field of mass spectrometry, we can use shotgun or discovery traditional modes, which, you're able to really quantify thousands of proteins based on their peptides. And we do digest proteins into peptides for mass spectrometry so we can get increased accuracy of mass. There is other methodologies being developed where we just look at protein intact mass, but that's still developing, and I will not talk about that today. On the far right side, where we have targeted methodologies, these are methodologies that allow us to very specifically monitor one or more proteins, but the numbers are much smaller here, maybe 100 proteins. ELISAs are the most common methodologies, or things like Western blots, where we're being able to quantify and target specific proteins. On the mass spectrometry side, we do the same thing using a technology called multiple reaction monitoring or selective reaction monitoring, where we essentially exclude all the peptides except the ones that we want to measure, and those particular peptides we want to measure are those which are unique to specific proteins we're targeting. So essentially in this methodology, we exclude all the peptides, focusing on those specific peptides which are prototypic and representative of the protein or the peptide that has our potential post-translation modification on it. That's the exact opposite to the discovery methodologies, where we're trying to really capture every single peptide to our best ability. And as I mentioned, shotgun discovery has been around for over 20 years and has been very successful. Most recently, there's been a move towards a slightly different combination approach, which is, again, both discovery, but brings in some of the aspects of targeted. And this is called data-independent acquisition. And if you're using the triple TOFs, the trademark would be SWATH. In this way, what we do is we pre-build a library of all potential peptides that we would want to track. But we don't necessarily want every single peptide as we do in discovery, but a selective set of peptides which are representative of those proteins are in our library. So in that way, it's very similar to targeted. We pre-select the peptides that we want to really focus in on, but instead of just doing 100, we're going to be doing thousands. And then we pre-build this library. And then when we're running our data-independent samples, our samples on using data-independent acquisition, we're able to map those back over to our library and more efficiently see and quantify the proteins and represented by those peptides that we've targeted already. But again, many, many proteins. So if we look at this schematically, this is a protein, and as we said, we digest it often with trypsin, making peptides that have lysine at the N-terminus, at the C-terminus, or an arginine at C-terminus. And that peptide in discovery mode, we're looking at all of those peptides, and at the bottom, each one of those blue triangles is supposed to represent a peptide that's in our sample, and as they get injected into the mass spectrometry over time, we detect them. And the one that we're interested in is in red, and it's coming out and being eluted into the mass spectrometry at a specific time. In MRM assays, essentially we exclude all those blue peptides and focus in only on the red one, and we're able to detect that peptide. And the way that we know that it's that specific peptide is we give it additional energy in ms to the 2, breaks it down to smaller peptides, which are representative of the amino acid sequence. And these are called transitions. They are smaller peptides of the existing parent peptide. And that's represented on the far right, where we can see the blue peak area, that's your parent, and all the breakdown peptides co-eluting right underneath it. And essentially when we want to quantify a peptide, we would quantify the peak area underneath that blue peak. But we know that it's the right peak because of all these smaller peptides, which are essentially, again, telling us the amino acid sequence of that particular peptide. So if we are now monitoring more and more peptides, of course, we want to not just quantitate on one peptide, but many peptides within our protein. They will elute at different times in our during discovery mode, and in MRM mode, we know exactly when they're going to come out, and we schedule them and essentially pick them up, and then we quantify again on the different peak areas of each peptide representing our proteins. And you can see how this is easy if we're only necessarily looking at one or two proteins, but when we want to be able to quantify hundreds of proteins or thousands of proteins, this becomes more complicated. So when we talk about citrullination, you should be thinking, well, why don't we just quantify those particular peptides that have citrullination in the arginine's been modified to citrullination. Well, the challenge is in mass spectrometry is that citrullination is still a very low abundant and has very low stoichiometry at the peptide level. So it's going to be very low in our sample. We have no direct enrichment strategies right now, so it's not like we can go pull out those peptides which are citrulinated. The citrullination itself is only a 1 Dalton mass shift, and if we have multiple charges on our peptide, of course, we're measuring m over z, so we're going to have even a smaller change. There are other post-translation modifications that have the exact same mass, so it's going to be important to take that arginine to citrulline change in our peptide to unambiguously identify it, and there's other parts of mass spectrometry that makes it more challenging. So what we did with the development of SWATH is to be able to think if we were able to enrich our protein library with citrullonated peptides, maybe that would help us see those citrullonated peptides when we moved forward to large-scale monitoring of this protein modification in complex samples. So what we did is we take our protein mixture, let's say it's of the heart or an aorta, we pre-citrullinate it, so we maximally citrullinate it by adding the enzyme that induces this. Digest it. In this case, we use lyc because we're changing arginine and we don't want to use trypsin, which can cleave. After that, we want clean cleavages, so we use lyc. We digest it. We make a peptide mixture that's now heavily enriched of all possible citrullonated residues. We now include this into our protein library. Now, we actually do a few more things. We noticed while we were doing this that when you have a citrullonated peptide, it will change its retention time on an LC by up to or more than 5 minutes. So we have created an algorithm that allows you to help with further characterization of that characteristics that are unique to a citrullonated peptide to unambiguously identify and confirm that you have a citrullonated peptide. And when we did that, if we were, for example, to take a heart and pre-citrullinate it, digest it with lysine, make our peptide library, and then run heart samples from human hearts in DIA mode, and then look at those and compare them back to our library, add in those other additional characteristics to ensure that we really unambiguously identify those citrullonated peptides, we can drastically enrich our competency and the number of peptides that we are able to identify. So what you see in the bar graph is if we were just to take a sample and run it under shotgun, we would manually have to go through every single one of our spectra that's being called to be citrullonated because of these other issues. So you might have 1,000, and you will end up with being able to identify about 9. When we made the library, we were actually able to enrich about 483 additional peptides that are citrullonated in the heart, and we put those into the library. So that's a maximum number we should be able to identify. And when we use our new pipeline, where we're essentially running the DIA and then putting it through our algorithms, we're able to identify and quantify 304. So this is a huge ability now using DIA methodology and this new algorithm to be able to confidently identify citrullonated peptides. So when you do that in heart, and we did it against both healthy human heart and heart that was obtained from individuals who have two different types of heart failure, ischemic and dilated heart failure, and asked the question, are there citrullonated sites or proteins which are differentially modified when you have heart failure? So of those 304 citrullonated sites that we found in human heart, 145 of them were different, 52 of them were differentiated. 52 of the sites were different between control and the two forms of heart failure. And what you see in the network analysis are the types of proteins which are enriched in citrullination that were changed in heart failure. And you can see that there is a lot of mitochondria proteins, proteins associated with the proteosome, nuclear regulation. That's something that has been known in other fields looking at citrullination, that the epigenetics can be controlled and modified by citrullination. But in the heart, we had this subproteome at the bottom called myofilament. And this is enriched, again, in heart. This is the contractile proteins that are responsible for heart contraction. But the myofilament proteins were also one of the subproteomes that were enriched and altered with citrullination in heart failure. So contractile force is produced in the heart, and that's what allows it to contract and relax and push the blood out into the body. And up in the top are pictures really of an isolated cardiac myocyte. And this was actually done in a collaboration with David Cass and Jonathan Kirk, and they did this beautiful set of assays to be able to figure out in an isolated cardiac myocyte whether or not we would change the force production of the myofilament proteins when those proteins were citrullonated. And that's what you see on the left-hand side. So what you do is you take an isolated myocyte, you remove the membrane and all the soluble proteins, and you're really left with the myofilament. And as you add increasing calcium concentrations, those proteins will contract, and you can measure them, and you have an increase of force. When those myocytes were citrullonated, so when we treated with LIMPAD, we were able to shift the calcium sensitivity. And that's actually very important because in heart failure there's two major parts that happen. One is you get a decreased force, and we have many mechanisms that we know about in our field that does decrease force. But what's been missing is really the ability to figure out a mechanism that will change the calcium sensitivity. And that's the other part that happens with heart failure. You require more calcium in order to induce any force at all. And so citrullination obviously changed force by the calcium responsiveness. And on the right-hand side, what you see is a schematic of the myofilament proteins. You have the thick and thin filament, the thin filament being composed of actin, tropomyosin, and the troponins. And then you have the thick filament, which are the myosins. And also you have the M and Z line proteins, which help to essentially align the myofilament proteins and keep them in alignment. And what is color-coded is those proteins which were citrulinated. So in black, almost all the proteins labeled, well, all those proteins labeled in black are citrulinated proteins. In green are those that should change with ischemic heart failure, and in red those which were changed with a dilated heart failure. So both heart failures were modified, and there were specific proteins within the myofilaments that were more modified compared to control healthy hearts. So when we stepped back and thought, wow, this is the first time anyone's shown that citrullination can happen in the heart and that can change contraction, and perhaps this was a mechanism forth to understand how we were changing calcium concentration or calcium sensitivity in heart disease. But as I mentioned earlier, what's interesting is that citrullination can also be autoantigenic. So we came forth with this kind of bigger hypothesis that in the heart itself, during heart failure, you will have citrullination happening, including to proteins like cardiac troponin I, which is already a circulating biomarker that when you have a heart attack, you will release or increase your release of cardiac troponin into the bloodstream. We monitor it, and if it's super elevated, we know that you're having a heart attack. So we thought, well, if this is happening in heart failure, which we know there is continuous leakage of cardiac muscle proteins into the blood, and if it's modified and if you have citrullination, perhaps if it's long enough or you have severe enough heart failure, perhaps you are actually releasing the modified form of troponin I, and thus could be an autoantigen, and it could drive actually an autoantibody response. So we have been proposing that with heart failure, as you progress and you're having more citrullination of our proteins, and if they get released as they will with heart failure, you could stimulate an autoantigen response and an autoantibody response that would then drive and make your progression worse. And perhaps the difference between how individuals progress with the disease is not just what's happening mechanistically within the cells, but also inducing an autoantibody response. So we went after it, this concept, by taking heart tissue, fractionating it extensively into about 500 fractions, arraying them on glass wells. And so now what we've done is we've taken hearts from normal as well as from ischemic and dilated heart failure, fractionated them, arrayed all of those fractions, which will contain anywhere from 1 to 200 proteins, and then started to screen serum against these proteins. And these proteins, remember, will both be unmodified as well as citrulinated. So as we screen patients which have RA, and patients which have RA but have heart failure or have increased calcium scores, meaning that they're at risk of heart failure, we were hoping to be able to find some autoantibodies being induced, and they would bind to the array and we'd be able to detect them. So that's what happened. And here's two examples. When we did this, some of the spots kept coming up as being potential autoantibodies or proteins that are autoantigenic. So we analyzed by mass spectrometry those specific spots, and cardiac proteins, which were citrulinated, were found in those fractions. Two of them with myosin and troponin. And so what we did on the second one is we took purified myosin, human myosin, and purified human troponin. We took them and we citrulinated them and then spotted either the unmodified or modified forms of myosin and troponin, and then ran the same sera back over them. And that's what you see on the right-hand side. You can see that, especially in the bottom with troponin, that the red, which is troponin when it's modified with citrullination, has a large response. So this is the autoantibody response. So autoantibodies in these different individuals that have RA are binding to the troponin that's modified, but not the unmodified form. So in other words, this is some of the first indications that modification by citrullination of myofilament proteins, and we suppose other cardiac proteins, can be released into the blood can cause an autoantibody response, and we can pick that up using these arrays, using the citrullonated form of these proteins as bait. So now that's the reason why we want to care about citrullination, and being able to find drugs now that are able to modify and modulate the PAD enzymes are a way of going forward. Again, this was brand new data that was coming out, and citrullination had never previously been shown to occur in the heart. So how can we scale this, and how would you go forward to actually making the biomarkers? So to do that, we have to be able to do large numbers. And so in the pipeline of proteomics, you go from samples to being able to do sample prep, where we do this digestion. So essentially we do protein denaturation, reduction, alkylation, digestion using either lyc for citrullination or trypsin for normal protein quant. And then we have to desalt and get it onto the mass spectrometer, and then we need to do data analysis, either the targeted MRM assays or things like DIA, which was the example I just showed you with citrullination. And then, of course, we have to understand and get knowledge from that and move to more clinical assays. So we need a scalability. So over the last few years, we've been working with Beckman Coulter and SCIEX to develop this pipeline, really on the automation of sample processing, and I'll talk about that. And again, when you have increased sample processing, you get both an increase in time, so you're able to do it quicker, but also you should be able to improve the reproducibility, and the coefficient of variance should be driven down, and we hope driven down towards that of the level that you would get in a clinical chemistry lab. So this is Christy Hunter. She is from SCIEX. She was our partner in this, along with Mike from Beckman. And essentially, we used the biomarker NXP system, and we rigged it to be able to do essentially protein denaturation, reduction, alkylation, digestion on a 96-well plate. And at the bottom, what you can see is really the CV along the x-axis. So we're going to try to get it to be under 20% versus the peptides that we're monitoring. In this case, this is all the peptides in the sample that we'd be monitoring, which was hundreds. And you can see if we just do manual prep, which is, if you're only doing a few samples, you can be very, very good with this. You can have CVs around 10 to 15% if you're concentrating just on a few samples. As you start to increase the number of samples, of course, your CV is driven out. So when we talk about biomarker validation, we're doing hundreds of samples. Normally, even with the best people, we're hovering with CVs over 20%, maybe between 20 and 25%. And of course, some of that is from the LC response itself. And so with what we're trying to do is have our automation system to be equal to the very best manual preparation and to keep our CVs low. This is on two instruments. This is Chin Fu on our left side. She is from Cedars-Sinai. She leads our high throughput center, and she was working with Christy Hunter to develop these systems. So we have two NXP systems, biomarkers at Cedars-Sinai as well as at the SCIEX labs. And you can see as we're comparing day-to-day reproducibility that the two instruments are working very well. And this is sample preparation followed by MRM assays of many peptides, and again, being able to reach our CVs of well under 20%. So this is the protocol. We've reduced the protocol, and essentially what we do is we take plasma or serum, we add in beta-gal, that is our sample processing control. We put it as a protein level, and we're going to monitor it as 3 peptides at the MRM level to know how well our sample is processed, as well as our internal standards. For each of the peptides that we might be monitoring. We do protein denaturation, reduction, alkylation, trypsin, we quench, and then we do online desalting onto the LCMS, either the 6500 Q-traps or the 6600 triple toss. And essentially we now have the digestion protocols down to 2.5 hours, so you can do 96 samples in about 4 hours with reproducibilities less than what you get in the clinical chemistry lab. What this means, however, is that you're able to do large numbers of samples easier than we've ever been able to do it before. This is to show you the 2.5-hour protocol, and this is tracking 2 different peptides. You can see, in fact, we don't even need to digest for 2 hours. We can do it much more, either on the left-hand side is beta-galglycine buffer, and on the right-hand side is what it looks like if you're monitoring it in serum itself or plasma, and we can monitor both albumin as well as the beta-gal peptide. And you can see that we can get digestion in even shorter periods, probably between 30 minutes, and shortening down the total time required to do sample processing down even more. So online desalting turned out to be very important. Desalting still seems to be a challenge that we could go over. it seems to be still an issue. So what we did on the 6500 was to actually do a divert. So we're able to divert off the salt and then really have the clean peptides go into the mass spectrometer. And this not only reduced the time required to do the whole processing, but also was able to keep our CVs very tight. And this is an example of looking at beta-gal and human albumin peptides over many runs and able to show you that we're able to get either with online divert or online trapping our CVs to be around hovering below or around 10% over many, many, many samples. So this is the total pipeline. We do automation now using the biomarker. In about 2.5 hours, we're able to add the N15 labeled peptides after the sample processing. We do online desalting onto the 6500 and do data analysis. On the right-hand side, we're looking at peak area or the CV of the beta-gal peptides that were spiked into over 500 samples that were run in triplicate on the 6500. And you can see how tight the beta-gal peptides that we were monitoring, again, beta-gal being the protein added before sample processing, so it's going to really monitor the CV across the whole pipeline, are coming in around 4% or less than 4% over essentially what was a 5-day run, a continuous run on the 6500 to quantify this set of MRM assays. So it looks like we're able to do large scale accurately for MRM, and we are moving this sample processing to being able to do it on DIA, again opening up the possibility of being able to do hundreds of samples in discovery mode as well as in targeted MRM assays. Of course, the big difference is the time of MS runs and the number of proteins and peptides you're measuring. But we're still needing to have good analysis of RA and cardiovascular risk. So in a collaboration with Joan Betharn, Adam and Mike and Nick, we went on several years ago to try to find biomarkers that could be very helpful to be able to predict if you have RA, if you're going to have cardiovascular disease. And so there was a large number, about I can't remember if it was 60 or 70 antibodies pairs for ELISAs that were analyzed, and a few examples are listed there, as well as our lab was responsible for developing MRM assays, and then we analyzed them on several cohorts. And this is our MRM assay. At the top you can see all the peptides we were monitoring, and at below this is actually the setup of how we blocked all the samples and all the QC that we ran. So we ran standard curves at the beginning and end, and we're going to be, again, this is going to be about a 5-day mass spectrometry run, and we essentially had cases and controls. We randomized those blocks, and every group of samples we would run a short QC run, which is essentially a 3-point standard curve along of it. So we're able to look at both the beta-gal across all of those samples that we were run, which is what you saw with CVs around 4%, but we can also look at all the peptides which are being monitored at all of these QC runs across the 5 days. And this is what you can see here. At the top you can see the beta-gal curves around the 4 days, the CVs of the 2 peptides, the red and the green. You can see they're very, very stable, all under 5%. And below are now looking at one of the proteins being monitored by 2 peptides and 2 transitions in each peptide. That's why there's 2 measurements per peptide. And you can see the 3-point concentration curve for each of the QC over the 5 days and how reproducible this whole pipeline is, both at the digestion level as well as the mass spectrometry. And this is looking now at the QC curves for 3 different peptides. Again, at the top being measured by 2 peptides, 2 transitions. Peptide 2, again, is 2 peptides, but the first peptide has 3 transitions we're quantifying, and peptide 3 has the third protein has 3 peptides which are being quantified, peptide 1 and 2 by 2 transitions and peptide 3 by 3 transitions. And you can see how beautiful the reproducibility is. So what we're doing now with the NPX, we're able to run large scale, both for MRM as well as for DIA discovery mode. And in this experiment, essentially what we did is we ran 4 samples per day, and we did the 2-hour trips in, and we're monitoring 70 proteins based on 190 peptides over 600 transitions. And all you need to take from that is that we were able to replicate these incredibly tight CVs across the whole protocol. Again, this is from the sample through the whole sample processing, the addition of the internal standards, running of the samples through the mass spectrometry, really on these large MRM assays. So let's take a look at that data. This is looking again at 3 NPX runs, so 3 different days. So this is looking for intraday variability, and you can see how beautiful for that many proteins and that many transitions we're able to get under 20% CV. So this opens up the door of having discovery, having the same CV quality as you have for clinical chemistry assays. So both for discovery and for these targeted, being able to have the accuracy in proteomics that are really as good as the clinical chemistry labs. So that's where we are right now. So what's next? Can we use this technology and push it even further? If you can do 500 samples, why can't we do 1,000 samples? Why can't we do 5,000 samples? Why can't we do 500,000 samples? If you start to talk about moving in that direction, now you can start to talk about patient-centric health screening at the population level. So this is kind of going to where the future is, and we hope the future will be very quick. So what we can do now is you can take blood spots, so you can prick your finger and put it on filter paper and send it in and analyze by mass spectrometry lipids, small molecules, and proteins. Obviously higher abundant proteins in the blood. We've been working with a company that has a volumetric microsampling device called Mitra. This is a company called Neoterix, and we've been working with them for a while to be able to determine how we can use this volumetric micro device to sample blood from a finger prick. And they have 2 forms, a 10 microliter and a 20 microliter draw, and this is very precise. Excuse me. You can dry them and then in theory, ship them to us, and we can do mass spectrometry on them and monitor proteins. So we started this project, and no one had done that yet on the Mitra device, gotten proteins out of the device. So we're going to develop that pipeline. So why is this important? So right now, there's a lot of drive being able to use device monitoring of patients. So Fitbit, being able to use your phone to be able to monitor heart rate, and these devices can go out to the population, and you can understand a lot about the physiology and the pathophysiology phenotype by using these devices. But what's been missing has been the quantitation at the molecular level. And so if you're able to use these devices that you can prick your finger, accurately draw some whole blood into that, mail it to a research lab or to a pharmacy, that was my dream, is to have this in the pharmacy, to be able to then quantify proteins that can tell you really your health status. And then you can have very fast feedback to find out and monitor whether or not you're taking your drugs, whether or not your disease is progressing, whether or not you're healthy, or if there's some issue with your health. If you can do that, if you're able to send these devices out, you can reach all populations. You can reach populations who are not close to a healthcare center. You can reach populations that do not want to come in to see a doctor, and you can also monitor those patients that are coming out of a hospital or on long-term care. So there's a lot of potential if we're able to collect blood out in the community, send them in, and get accuracy. But of course, what we're talking about is large numbers and the need to be very accurate, and also having the right biomarkers to be able to monitor it so that you could actually tell something at the molecular level. So the concept is with these Mitra devices is that is to be able to tick your blood and, whoops, for some reason my screen, okay, there we are. So to finger prick your blood, draw your 10 microliters, you air dry them, and you send them to us to do it. So again, what we need to be able to do is we need to be able to extract from these Mitra device tips to be able to do denaturation, reduction, alkylation, digestion, cleanup or desalting, and we have to have the right MRM assay to make sense of what we're looking at, and then to be able to quantify it and then be able to get that information back to our patients. So these methods are able to quantify any peptides. So these, again, can be peptides that are specific to an isoform. They can be specific to a post-translation modification. So if you have a citrullination of an arginine residue and you want to measure the concentration of that particular citrullinate peptide, what you do is you make an MRM assay to both the citrulinated peptide as well as its parent peptide that is unmodified, so you would be able to get a ratio of modified to unmodified, as well as, if you want, you can have other peptides which are unmodifiable from the same protein and get a total protein quantitation. So that's the beauty of mass spectrometry and something that you cannot do with ELISAs. With ELISAs, what you can do is you can only measure one epitope. So if you wanted to measure a modified form, you would have an ELISA to the modified form and a different ELISA, which you would have to run separately for the unmodified. In MRM assays, because we can multiplex so well, we can have in the same multiplex peptides that represent the unmodified protein, so you can get total protein concentration. And then for that very specific site that you want to measure, that citrullonated site, you can have a peptide that is to the unmodified peptide as well as to the one that's modified. So you can measure the citrullonated peptide, the equivalent unmodified site, plus these independent peptides. So you could have multiple measurements that are all run at the same time in the same multiplex. You can do that for phosphorylation. You can do that for, which we've done for cardiac troponin I. We published a paper several years ago where we tracked in a single MRM assay over 12 phosphorylation sites, as well as the total protein concentration of cardiac TNI based on a 13th peptide. And we were able to really get accuracy and measurement of every single phosphorylation site of that very key protein, as well as total protein quantitation, and for the first time measure independently each of the phosphorylation sites. And actually the data that came out of that was quite unexpected and exciting for the field, that there was brand new phosphorylation sites no one knew about before, and they were the ones that were changing more in heart failure than the classical sites that people had been measuring and focusing on. So it really changed that field from focusing on a subset of phosphorylations to a brand new set, which we think are driving heart failure. So this is the type of things you can do with MRM assays. And it's also true whether or not you're doing it in a clinical lab, whether or not you're doing it for research, or doing it like we're talking about here on the Mitra devices, where we're trying to do it on large scale. You can get the specificity of the disease by honing in on those disease-modified forms and quantifying both the total protein concentration as well as the modified form itself in a single assay, which is essentially so exquisite. So the way that we needed to be able to move from our traditional NXP automation work system to being able to do it on the Mitra device took some work. It was done by a wonderful postdoc in our lab, Irene van der Broeken, and essentially these are the changes that we did from our normal to the Mitra device. And the big difference is that we had been working in plasma, and now we had to work in whole blood. Worked very well, and here is an example of the MRM multiplex that we used on Mitra, and you can see the peptides are representing the different proteins. And this is a great example that if we had a modified form, for example, in beta-gal, and let's say this was the modified form, we could measure it plus a peptide, its unmodified form, plus peptides that were to different parts of the same protein, and obtain both the modified and unmodified concentrations in a single multiplex, because these peptides behave very differently and will be eluting at different times. And again, in MRM, we exclude all the peptides from the mass spectrometer except those that we're very interested in. And so what we did here is you can see we were looking at putting the Mitra devices in, extracting the proteins from the Mitra device, denaturation, reduction, alkylation, digestion, desalting online to the onto the Q-traps, and then analyzing each of these proteins. And what I'm just showing you here is some of the data for the apolipoproteins, A1E, that we're measuring ApoE with 3 peptides, ApoC1, ApoC3, and CRP. CRP is an interesting protein. This is a protein that is very good for assessment of the inflammatory status of individuals. So it's C-reactive protein. We're measuring it from one protein. You can see it's a very low abundant peptide. It's let me see if I can get the arrow to work. You can see it right here. It's a very low abundance, but it's very clean in the mass spectrometry because we exclude all the other peptides, so we can assess it. And also cystatin C, and cystatin C is a protein that is measured really as a way, or it's a new marker that's coming up for being able to measure kidney function instead of creatinine. So cystatin C, CRP, you're going to be able to measure kidney function. CRP is going to give you inflammation status. Of course, the apolipoproteins are surrogates for your lipid panels. And so already we're able to, just on these proteins, able to assess an individual based on several of the key parameters for wellness. And essentially, when we did that, if we were able to do an N of 6 and then again over 3 days, you can see how tight the coefficient of variance are for these particular peptides. And remember, what we want to be able to beat is that 20% CV that is done in the clinical chemistry labs. I must clarify that in clinical chemistry lab, many of the assays are better than 20%, but that is really the cutoff. We need to be under 20% in order to have the same quality assay. So where are we and what did we talk about? Well, what we talked about today is the ability to get at mechanisms, and sometimes these are new and unexpected mechanisms, such as the modification citrullination in heart failure, which had not been known until just really recently in the last 2 years, and the fact that the citrullination can impact contractility, at least it looks like it can based on research studies. And that means that we could have a mechanism of being able to maybe inhibit and block the change in calcium sensitivity in heart failure, which has been today not been possible. We don't have any specific pathways to target to do that. We were able to do that by using SWAT DIA acquisition in a new and interesting way by pre-building and pre-enriching our library so that we are able to more efficiently identify those peptides that are modified. We came up with the hypothesis that this modification, which can be autoantigenic, could in heart failure being leaked out into the blood and produce autoantibodies, and we have some preliminary data to show that this could be the case, especially if you have RA or at risk. We showed that being able to have larger numbers by having very efficient and robust sample processing linked either to MRM assays, these targeted assays, or to discovery. We're able to be able to increase our numbers both for discovery as well as for MRM assays. That this really, when you have the pipeline well, you can have very good CVs well under that of clinical chemistry requirements, and we are able to monitor and assess proteins over long periods of time and keep that very consistent. And then the last part to talk about this concept that we're able to use mass spectrometry, because again in 10 microliter draw you can't be running ELISAs, you would only be able to look at 1 or 2 proteins. But with mass spectrometry, because we can multiplex many peptides together, we can multiplex and monitor and quantify many different proteins, or many proteins with their modified forms in small amounts of samples. And if we're able to link that up with microsampling devices that can be done in the pharmacy or at home or wherever you are, that are safe and effective to be sent into a lab, or we can monitor it, we should be able to effectively monitor, at least now, how your kidney function is going, your inflammation, and your equivalent to your lipid panel. Are we doing that yet? No, but we're certainly on the way, and that is what the future holds as promise to be able to do. So I have an amazing lab. I'm very fortunate, and in red are those individuals that contributed data and thoughts and towards today's presentation, as well as we have wonderful collaborators both at Hopkins and in RA, Joan, Adam, and Mike, as well as we're very fortunate to work with different industry partners to have access to these different mechanisms, both the automation of the sample processing for mass spectrometry, and also Neoteryx for working with us to be able to work on the Mitra devices. So thank you. So if you have questions, I'd be delighted to answer any. And again, I wanted to thank you for spending some of your day with me.
Thank you, Jennifer, for bringing us that informative presentation. Before we get started on our question and answer session, I do want to remind our audience how to submit their questions. Questions can be submitted by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of your questions as we can today. Our first question, an attendee asks, do these methods allow us to identify if all sites of post-translation modification were modified in vivo? What is the difference in interpretation when modification is done in vitro and then followed?
So what a great question. Thank you for asking that. So first thing, traditionally when we're doing shotgun for something like phosphorylation, we attempt to see all the phosphorylation sites, but you don't. The difference with the approach that we're using, where we pre-make the library, where we on purpose try to induce 100% of that modification and put that into the library, so if it's there we can see it, we increase our probability of seeing it. And that's the advantage of this approach of pre-building a selective library where we've really enriched it, hoping to be able to pull out those very low abundant peptides that have that modification, because we know what we're looking for. So if you know what you're looking for, you're more easily able to see it than when you go hunting for it. So that's the approach. So do we see them all? Yes, within the limit of what our mass spectrometries are able to detect. So we have a dynamic range of a mass spectrometry. It has, from the highest abundant peptide to the lowest, there's a concentration, so gradient. So if it's within that dynamic range, we can see it. It's equivalent to an ELISA. With an ELISA, we talk about a lower limit of detection and a lower limit of quantification. We have the same thing in MRM assays and the same thing in DIA and discovery mode. We have a lower limit of detection and a lower limit of quantification. So as long as our peptides are within that, in our mass spectrometry, we are able to quantify it, and really in the same conceptual way as you do with an ELISA. But of course, if a modification is below that detection level, we won't see it. So it could be there, and we won't see it. So in mass spectrometry, just as it is with an ELISA, an absence to detect something does not mean it's not there. It just means it's below our detection level. But if we see it and are able to quantify it, we're pretty sure it's there. There are some caveats, both with ELISAs and with MRM, where that isn't always the case, but because in mass spectrometry, we're also measuring these transitions, these breakdown products of the peptides, we have additional information that makes it so that we can be pretty confident that we're measuring the correct peptide. And because often we're measuring more than one peptide, we can be pretty sure that we are measuring multiple measures of the same protein. So it's very good. So the question is, what's the difference between in vivo and in vitro? So when we're doing it in vitro, we're trying to maximally modify it. That's our purpose. We're trying to see them all. Because in vivo, of course, that may or may not be the case. And what we're trying to do is to quantitate each different site and find out how they're varying within an individual. And some individuals, site 1 may be varying, but site 2 always happens. And so we're trying to find that site 2 that's always happened, that common mechanism, that common site. We don't want to know just if a protein's modified. We want to know the actual site, because we find it's unlike with citronation, the example I showed us, when there was actually different proteins that were being citronated. Often, and especially with things like phosphorylation and oxidation, it is a different residue within the same protein that's modified, and it's knowing that specific residue that's important both for the diagnostic as well as for the function and for the therapeutic. So thank you. That was really a wonderful question.
I see. Great answer as well. Our next question, does discovery or targeted proteomics approaches warrant automation? If so, how big of a lab or core?
So that's also a great question. So I believe that we need to have very accurate measures, whether or not we're measuring proteins for discovery or measuring proteins for targeted MRM and biomarkers. Biomarkers, everyone affirms that we must have that. We must equal that quality that we expect from a clinical chemistry ELISA, so 20% CV. But I would say to you, if you're using automation, there's no reason that you can't have that with discovery, and especially in post-translation modification, where small changes can have drastic effect on function. For example, the protein myosin light chain 20 is the protein that gets phosphorylated when smooth muscle contracts. So if 20% of that protein is phosphorylated, you get full contraction of smooth muscle. So we need to, as proteomics people in discovery or in targeted, to be able to quantify 20% of that protein being phosphorylated at a specific residue. Now, if you're not having sample processing that allows you to measure with 20% accuracy, you can't. You cannot measure that change. And that change in myosin light chain 20 is the most important phosphorylation in smooth muscle, because essentially it's making it contract. So protein changes and protein changes at the post-translation modification often is not huge. It's only 20 or 30 or 40% of your protein is going to be modified. And if that modification is at a binding site, that can change completely how your cell will respond to a signaling molecule. So I would say that everyone, whether or not you're doing discovery or targeted, needs to be able to do accurate measurement, and certainly automation of sample processing allows you to do that along with standards. But equally, if you start to automate, all of a sudden you can add more samples. So instead of doing 10 samples, you could do 100 samples. If you could do 100 samples, you could do 1,000 samples. And so I always push back to people, including my own lab who does research, and say, why are we doing 20 samples? Why aren't we doing 200? If it really is that much quicker to be able to do sample processing. If you can do 200 samples, think of all the perturbations and experimental conditions you could do in a single experiment. Think how much more understanding we would have of biology if we were able to just increase by a factor of 10 the number of experiments we're able to do. So I think small labs and big labs should be moving in this direction. If it's a series of small labs, then you could share an instrument, a processing system. But yeah, I think small and big labs, I think in proteomics, really need to move to automation of sample processing and with inclusion of standards in order to track the quality of your experiments and move your numbers to larger to really help proteomics do what we've been promising it could do for a long time.
Okay, great. Our next question. From the identification of rogue proteins, have any tailor medicines been made?
Oh, what a wonderful question. I wish the answer to that would be yes, but oh, excuse me, sorry, I have a bad cold. But to date, no. Mainly because proteomics and the identification of these molecules is only coming on at the scale it's needed in the last few years. But there are biomarkers, such as cardiac troponin I, which the phosphorylation modifications really drove and helped refine that particular assay that is now the most common assay used worldwide for really determining whether or not you're having a heart attack. Two new markers that are going to be coming out around traumatic brain injury, where we know both the markers that are released with traumatic brain injury, these proteins we can use for diagnosis, but they are also citrullinated. And when they're citrullinated, we believe that they're producing autoantibodies in those severe individuals who are injured. So these assays are coming on board. We are working with pharma to help them get their drugs on target by monitoring the phosphorylation status as they do perturbations with different drug targets to a specific pathway that they want. This is coming online. So proteomics is now really impacting it. So expectations is that we are helping to refine already proteins, and in a collaboration again with David Cass and Jonathan Kirk, that was published last year on protein kinase G, was a perfect example of how proteomics was able to help them define this kinase's pathway, that there was two different pathways depending on how you were stimulating and whether or not you had heart failure or not, to help those companies who are producing inhibitors to target really the right protein. So that's also a great example, but it's not yet in the clinic, although clinical trials are being planned.
Okay, great. I believe that is all the time that we have for questions. Do you have any last comments or advice for our audience?
Just please, I hope you embody and embrace proteomics and the scalability that many of our labs, not just our labs, many labs have been working hard over the years to be able to really get it to be reproducible and robust, to be able to do population scale proteomics. And we're at that point now. Are there still lessons to be learned? Of course. But the exciting part is that there's so much more that we can do now, both at the discovery, at the MRM level for really remarkable quantitation in the clinical chemistry arena, but also in the population arena with these new devices coming out. So again, thanks everyone for spending a part of your day with me, and I hope that you enjoyed it.
Thank you, Jennifer. It was a wonderful presentation, lots of great questions from our audience. To that end, if we were not able to answer your question today, we'll definitely be following up with you via email to get your questions answered. I'd also like to thank Beckman Coulter Life Sciences, our sponsor today, for making this educational webcast possible. Today's webcast will be available for on-demand viewing through October of 2016. You'll be receiving an email from LabRoots alerting you when this webcast will be available for replay. And we invite you to forward that announcement to any colleagues who may have missed today's live event. Thank you again for being with us. We hope we see you next time. Goodbye.